Okay, chapter 10 is about the characterization of nucleic acid and protein. And the first of all, the nucleic acid, right? Nucleus, they can degrade the DNA by breaking this phosphodiester bond, right? And there are two kinds of the nucleus. One is called endonucleus. The other one is called exonucleus. Exonucleus can degrade the DNA, right, at the end of the DNA molecule externally, and endonucleus will degrade the DNA internally. They will break those phosphodiester bond internally. Okay, they hydrolyze the internal bond, and when the nucleus hydrolyzes the ester bond in the phosphodiester, right? This is a phosphodiester bond, the ester bond, right? They have a specificity for either the ester bond, and then they generate 5' nucleotide or the 3' nucleotide. And exonucleus, they can attack the polynucleotide chain from the 5' end and hydrolyze the 5' to 3', or attack from the 3' end and hydrolyze 3' to 5'. And the endonucleus attack the phosphodiester bond from the 5' end or from the 3' end. Okay, so they can attack this phosphodiester bond at both the 5' end and 3' end. And there's some nucleus, all right? They are specific for the single-stranded nucleotide strand or the double helix strand or both. And here's just the exonucleus example, like a BAL 31, all right? So you can see they can degrade both strand, all right? 5' to 3', 3' to 5'. Okay, they degrade both strand. And exonucleus 3 can only degrade single-stranded, and that from the 3' end. Okay, that's the exonucleus. They degrade just one strand, all right? And then a different type of the endonucleus. So like S1 nucleus. S1 nucleus only cleave the single-stranded, okay? And the DNS1 can cleave both single-stranded from the nick here, or this single-strand, or double-stranded. S1 nucleus can only degrade this single-stranded. DNS1 can degrade both single-stranded and double-stranded. And then a recession endonucleus, which is very special one, very important one. Here, recession endonucleus, they recognize a specific nucleotide sequence and cleave the DNA internally. Okay, that's the recession endonucleus. And the initial observation of this recession endonucleus is the discovery of recession endonucleus is because the scientists, they discovered that the bacteria that resistant to the viral infection, and then they realized those bacteria they produce a specific enzyme, the protein enzyme, the recession enzyme. They can recognize the viral DNA and then degrade the viral DNA. And so that's how they discovered the endonucleus. And how they distinguish the difference between the viral DNA and the bacteria DNA. That's because the viral foreign DNA are not methylated, and those bacteria DNA are methylated, so that the recession enzyme, they will not digest those methylated DNA. Instead, they degrade those viral DNA. So it's a kind of protection defense mechanism, right? The recession endonucleus they discover in the bacteria. Okay, And there are 3 different types of a restriction endonuclease. Okay. Type 1, okay. Type 1, type 3 are complex and only a very limited application in the biotechnology field. Only the type 2 is very important. Okay. And type 1 enzyme normally their recognition site and the cleavage site is about 1,000 base difference. And type 3, their recognition site and the cleavage site is about 24 base pair difference. Only the type 2, okay, the type 2 restriction enzyme, the cleavage site will be exactly the same as the recognition site. So for example, like the EcoR1 enzyme, they recognize GAA-TTC, all right, and then also the complementary strand, also GAA-TTC or the hexamer, all right, palindromic sequence. They recognize these, and then they also they cleave here, right between G and A here, all right. So the recognition site and the cleavage site is exactly the same location. And ALU1 recognize AGCT, and they cut here between G and C. So this one, after they cut, they produce this sticky end, right? This is so-called sticky end, and for ALU1, they produce the blunt end, all right? And PSD1, they recognize CTG, CAG, CTG, CAG, and then they cut between A and G, and then they dissociate these 2 molecules, that's sticky end. And this sticky end is called a 5' overhand. And this sticky end, all right, it's called 3' overhand, because this 5' sequence here is single-stranded. 5' here is single-stranded, so it's 5' overhand. This 3' overhand, the 3' end here is a single-stranded. 3' end here is single-stranded. So it's 3' overhand, and this is blunt end. Okay, that's restriction enzyme. So this restriction enzyme, isolate from different bacteria. They recognize cut DNA at the same site. We call it isocytomer. All right, isocytomer from different bacteria recognize the same DNA sequence. That's called isocytomer. And then some are recognize and bind to the same DNA sequence, but cleave at different position. We call it neocytomer. All right, neocytomer. And other restriction enzyme, they produce the same nucleotide extension, but they have different recognition site. That's isocytomer. So that's 3 different types of enzyme. All right, we have isocytomer from different bacteria. They recognize cut at the same position, isocytomer. Neocytomer bind to the same sequence but cut at different positions, so produce different single-stranded extension as neocytomer. Isocytomer produce same nucleotide extension, but they recognize differently. Okay, that's isocytomer. And the restriction modification system. Okay, so almost all the restriction endonuclease are paired with methylase. Now recognize the DNA methylation site. All right, as I mentioned that the originally they discovered from bacteria, and the bacteria they develop these systems to protect themselves. Therefore, each enzyme they have the specific methylase that can recognize and methylate the same DNA, so that they can protect the bacteria DNA. All right, so that's why you have this restriction modification system. So RM system. All right. So the 2 enzymes, we have restriction enzyme and methylase. We call it RM system. Because after the methylation, the DNA sites are protected from the cleavage by the enzyme. All right. In the bacteria, during the replication, the 1 strand of daughter duplex is a newly made strand is unmethylated. And because the parental strand is still methylated, the half methylated, we call it hemimethylation, enough to protect the DNA duplex against the cleavage by the endonuclease. And then the methylase have time to find the site and methylate the DNA, right? To give the fully methylated DNA. This is a host defense mechanism in bacteria cell to protect their own DNA from restriction enzyme degradation. Okay. So just like this, after the replication is hemi-methylated, all right? But this still enough to prevent the restriction enzyme digestion. And so the methylase has enough time to methylate the other newly synthesized strain. So this is the RM system, okay? So the enzyme, then I will stop right here, okay? I'll stop right here. And then we will move into this laboratory, the restriction enzyme laboratory here. Analysis of restriction digester fragment, okay? So before sequencing a large stretch of DNA, some preliminary mapping is usually done to locate the cutting site and to examine the size of fragment. And a restriction digester can be performed in a microcentrifuge tube in the presence of the components like the magnesium, template DNA, restriction enzyme, okay? So a restriction digester results in the number of DNA fragments, and the size of the DNA fragment depends on the exact position of the recognition sequence for the endonuclease in the original molecule. And this restriction digest DNA fragment can be analyzed by gel electrophoresis, all right? And then we can, from those analysis, the gel electrophoresis, we can generate the restriction map, okay, based on the restriction site determined by the electrophoresis. All right, the restriction map can represent the linear sequence of a site at a particular restriction enzyme for the restriction enzyme, okay? So that's why the restriction digest mapping is important. So the procedure will be, first of all, digest DNA with restriction enzyme, and then you resolve the fragment by the gel electrophoresis, and then based on the number of bands, that can determine how many restriction sites. And also the size of fragment that can indicate the distance between the restriction site. Okay, so these are 4 important steps for the restriction enzyme mapping. And now this is the example. Okay, so the example, like for example, use restriction map to work out with the restriction endonuclease to obtain the DNA and containing the specific gene. All right, that's the one application. Use the enzyme to cut and to get a specific gene that you want, and the mapping, right? And to determine the unknown DNA sequence, all right? And the restriction mapping here. So let's say, for example, this is a lambda DNA, 49 kilobase long, all right? And then we have this blue is bagel 2, cut 6 different sites, 1, 2, 3, 4, 5, 6, 7, 6 sites. And give you this fragment, 1, 2, 3, 4, 5, 6, 7 fragment, okay? Because 6 sites. So for linear DNA, if you have 1 site, 1 restriction site, that will generate 2 fragments. For 6 sites, that will give you 7 fragments. And then bandage 1, 5 sites, give you 6 fragments. And then south to 2 sites, give you 3 fragments. And then this fragment, then you can map then on the linear DNA sequence here, all right, to give you this fragment, okay? So these are some application for the restriction enzyme digestion. Now we go to this practical question here. So for restriction enzyme mapping, let's say for the circular DNA, okay? If you have one bandage 1 site, you cut it, that generate 1 fragment, and then electrophoresis give you 1 fragment, 4 kilobase fragment. If you have 2 sites, okay, 2 sites that give you 1 fragment, 2 fragment, and 1 fragment 2.8 kilobase, and the other one is 1.2 kilobase. Then you see 2 fragments here. If you have double digestion, 1, 2, 3 sites give you 3 fragments, okay? And then these are the size of fragment. Okay, normally based on these geodesophoresis analysis, you'll be able to interpret the specific location of the enzyme. Okay, so let me see if we have the example here. Okay, here is the example. Construction the map. Okay, so this is the linear DNA map. Okay, so you have equal R5. This is the result. Don't look at the result here. Just look at the map here because that will be on the quiz on the final. Look at the geodesophoresis result. So you equal R5, you see 2 fragments. All right, for linear DNA, 2 fragments, that indicate there's only 1 cut site for equal R5. Bandage 1, 3 fragments. So that suggests that there are 2 bandage 1 sites. And double digestion, 4 fragments. So total will be 3 sites. To create restriction map, okay? So you just take either one of the single digestion first. Look at the, let's say we begin with equal R5. Equal R5, 2 fragments, all right, 1 site for equal R5. And then you can draw a linear fragment and put the, you know what, I'm going to turn on the camera. So I'm going to use my whiteboard, okay, so that you can see how I analyze the data, okay, because this will be on the quiz and the final or the exam. Can you see my whiteboard here clearly? Karen, you can zoom in. Okay, is this better? Right, is this better, okay? Do I need to stop sharing my screen or no need? This will be enough. This will be good enough. Okay, so to solve this problem, okay, as I mentioned, if you see the agarose gel, all right, you have this single digestion and double digestion. Okay, you have a single 1 and a single 2, all right, the 2 enzymes. And then double digestion, 1 + 2. To analyze, to solve the problem, you just take either single 1 first and then double second step. And number 3, you come to the single 2. And then you'll be able to construct a map. So for this question, so the equal R5, we will begin with equal R5. You see the 3.7, 2.3. First of all, 3.7 plus 2.3, so that's 6.0. So that means the linear DNA size is 6.0 kilobase, all right? 6.0 kilobase. And then you draw a line, a linear line, and then 2 fragments, so it's 1 side. You put 1 side here, okay? So that'll be equal R5, and then 1 side of the fragment will be 3.7 kilobase, and the other side will be 2.3 kb. Okay, let me get the eraser. All right, so we finished the first one, so I'm going to erase the first one here. Okay. And now we move to the second step. You look at the double digestion. Equal R5 bandage 1. Okay, that gives you 4 fragments, 2.5, 1.8, 1.2, and 0.5. Okay, 4 fragments. And here we have 2.3 and 3.7. and the equal cut here, okay? And then we have to find a combination, 3.7, 2.3. What give you the 3.3, 3.7, and which combination give you 2.3? So from here, double digestion, all right? So we can see these two give you 2.3, and 2.5, 1.2 give you 3.7, okay? So let's suggest these two cutting will be from here, and these two fragments from this fragment, okay? So now the next step is, let me get the different color. So that we have this one here, so that give you 1.8 and 0.5, all right? And here give you 2.5, 1.2, okay? And now I'm going to erase this second step here. And now you have to think different combination, different possibility, different scenario, because this could be this combination, all right? 0.5, 0.8, 1.8, or 1.8, 0.5. This could be 2.5 here, 1.2 here. Okay, so you have two different possibilities, and 2 multiply 2, so you have 4 different possible arrangement. Okay, 4 different possible arrangement. I'm going to list all 4 here. I use a black one here. 1.8, 0.5, and 2.5, 1.2. Right? And then we can have 2.5, 1.2, and then 0.5, 1.8. Okay? And then we can have 1.2, 2.5, 1.8, 0.5, 1.2, 2.5, 0.5, 1.8. So we have this 1, 2, 3, 4 different possible arrangement. Any questions so far? That's good. So and then to determine which one is the correct arrangement, then we go to the third step to look at the enzyme 2. So for the enzyme 2, we see the Banach 1 give you a 4.3, 1.2, 0.5. Okay. From these 3 fragments, okay, can you tell me which one is the right arrangement? That's correct. Number 3 is the correct arrangement. So how do you do this? So basically, from the number 3 step, you remove the other enzyme digestion. You look at the second enzyme. Okay, you remove the first enzyme, and once you remove the first enzyme, you see this combination is 4.3. So that means you remove this equal, you will see the inside here, you should have a 4.3, okay? And then on both ends, you should have 1.2 and 0.5. And then you will find 0.5, 1.2. And then in between here is 4.3. Okay, so this is how you interpret the restriction map. Okay. Then if we go to the next example here. Next example is the same. All right. Next example, we have 3 different enzymes. 3 different enzymes. I will explain the first one, then you should be able to know the second one. All right. So let's look at the SBA1. SBA1 give you a 24.5, and then XHO1 give you a 15, 33.5, and then for both XBA1 plus XHO1, that would be 9, 15, 24.5. To solve this, okay, go from this first one, number 2, number 3, all right? Number 1 is XBA1, give you these 2 fragments, so put it here, XBA1, so you have 24, 24.5, okay? And next step, look at this double digestion, 9, 15, 24.5, so that we know here will be the XHO1, all right? So that could be either 9-15 or 15-9, all right? 2 different possibilities. Now you look at the double digestion, single digestion here. Number 3 step, 15 is 33.5, so that you know 15, all right, must be here. And this give you 33.5. So that would be the answer. Okay? So that would be the how you construct the restriction map. Any questions so far? No, that's very good. You are very smart. Huh? Very good. Okay, so that's the typical restriction mapping. Okay, and that would be on quiz on the final as well. And now we will watch this video to perform the restriction enzyme digestion. Okay.
Restriction enzyme digestion is a fast and inexpensive way to gather broad sequence information about plasmid DNA. A restriction digest can give a generalized, bird's eye, or macroscopic view of the entire plasmid. It can also reveal useful information, like the purity and conformation of a plasmid, which is important for downstream applications, like transfection. For example, supercoiled DNA has higher transfection efficiency than nicked DNA. While in theory and in textbooks, restriction digestion is straightforward, interpreting the results of a digest in practice is easier said than done. In this video, we will digest a plasmid of known identity with restriction enzymes, run the products on an agarose gel, and analyze the results. We'll focus on identifying the bands produced under different conditions and use the results to make conclusions about the plasmid. Now, let's start our analysis. The first thing you should do is draw out your expected results. While this step may seem unnecessary, it's good practice and it will make your analysis go a lot faster. In this exercise, we would like to verify the identity of a plasmid we received from AdGene. We'll digest the 7.5 kilobase pair plasmid individually with either AGE1 or XBA1, which are both single cutters, and then with AGE1 and XBA1 together. We'll also digest the plasmid with EchoR1, which has 2 recognition sites in the plasmid. So let's draw out the expected results. Here's our gel. Lane 1 is the size marker, often referred to as the ladder. Lane 2 will be the undigested control. Because this plasmid is circular, the migration pattern of the undigested control diverges a bit from the migration pattern of the ladder, which was made with linear DNA. The uncut plasmid typically adopts a supercoiled conformation. That's because these plasmids were isolated from bacteria, and supercoiled is the native conformation of plasmids in vivo because it's compact. A compact conformation allows the plasmid to migrate through the gel at a faster rate than its linear equivalent. This is because it experiences the same amount of electromotive force, but less resistance due to friction. As a result, it runs a little smaller than the equivalently sized band on the ladder. This plasmid is 7.5 kilobases. So let's draw the supercoiled band right below the 7.5 kilobase pair mark. Another conformation of uncut plasmid DNA is nicked. DNA can be nicked naturally in vivo through the activity of topoisomerases. DNA can also get nicked during purification if the conditions are harsh. So it's common to see nicked DNA in your uncut control. When one strand of the DNA is nicked, supercoiling is released. So the conformation of nicked DNA resembles a floppy hula hoop or a hair tie, making it relatively large. In fact, nicked DNA is so big that as it's migrating through the gel, it experiences greater resistance due to friction than even the linear DNA, which can be more flexible than the nicked. Consequently, Nick DNA runs a little higher than the equivalently sized band on the ladder. Since we're still talking about a 7.5 kilobase pair plasmid, let's draw the Nick DNA a little above the 7.5 kb mark. Now that we've drawn our control, let's move on to our digestions. Lane 3 is the AGE1 digestion, which yields a single product. And because it's linear, its migration is consistent with the marker. So we expect to see a band at the 7.5 kb mark. Lane 4 is the XBAL1 digestion. Just like AGE1, it yields a single linear 7.5 kb band. So we expect to see a band in the same 7.5 kb spot. Lane 5 is the double AGE1 XBAL1 digest. According to our map, we would expect two products, a 7.2 kb band and a 300 base pair band. Since these two fragments appear in a 1-to-1 molar ratio, we know the 7.2 kb band will be 24 times brighter than the 300 base pair band, because it is 24 times as large. This means that if we don't have a bright enough 7.2 kb band, we might not even see the 300 base pair band. Lane 6 is the EchoR1 digest. EchoR1 cuts in two places, so we expect two products, a 5.4 kb band and a 2.1 kb band. Okay, this picture looks complete. Let's run our experiment and see our results. Run your gel long enough to get good separation of the bands. Don't rush it.
So here is just the standard agarose. You perform the gel electrophoresis. See the xylene sign up in the bromophenol group. And then examine the result with the UV box, ethylene bromide, then you'll be able to see the band.
Now that we've run our digestion, let's go through each lane and compare our experimental data with our expected results. We've marked the ladder with the sizes of the markers. Lane 2, the uncut negative control. We expect to see a supercoiled band and a nicked band, and that's what we see here. As expected, the supercoiled runs a little faster than the 7.5 kb band, and the nicked runs a little slower than the 7.5 kb band. Based on the relative intensities of these two bands, we can tell that the majority of our DNA is in the supercoiled conformation. Also, based on the sharpness of the bands and the lack of smear, we can deduce that we have a clean DNA prep. Lanes 3 and 4 are the AGE1 and XBAL1 single digests, where we expect a single 7.5 kb band, and possibly some of the uncut fragments. In each lane, we do see a linear 7.5 kb band. Lane 5 is the AGE1 XBAL1 double digest, where we expect two digestion products. We can identify the 7.2 kb band clearly. Since this is a new band, we know it must be the 7.2 kb digestion product. Looking for the 300 base pair band, we find that it's visible but faint. From our earlier calculations, we expect this band to be 24 times less intense than the 7.2 kb band, so the band intensity is not surprising. Lane 6 is the EchoR1 digest. Again, we see two new digestion products in this lane relative to the negative control lane. Notice we do not see any residual nicked or supercoiled bands, which indicates that the reaction was run to completion. Overall, our gel looks close to the expected results, and we don't see any bands that we can't explain.
So this is a typical research enzyme digestion process. And then after digestion, then you run the gel electrophoresis to confirm your result. All right. And from this, then you'll be able to perform these, okay, to construct a map. Okay. So let's say for today,